pass it on to you.
Okay, first of all, I'd like to thank the organizers. Okay, we're on now? Okay. First of all, I'd like to thank the organizers for inviting me. It's a pleasure to be here. And I thank Wenjing and Fong for inviting me, and I thank the students that have helped me out during my stay here. It's certainly an honor and privilege to be here this afternoon. I think my title was supposed to be The Future of Epigenetics or something like that. It's not. So what I'd like to do for the next few minutes that move? Okay. I'm going to give you a brief overview of why my agency supports, I'm going to give you an outline, first of all, what I'm going to do here. I'm not really familiar with how this is working out so well. But I'm going to give you a rationale for why my agency supports epigenomic research, and I'm going to give you an introduction, an overview of things that apparently, from what I've heard, you know a lot about already, but that would be the roadmap, epigenome program, some things about ENCODE, some things about the International Human Epigenome Consortium, the 4D nucleome, and a little bit of overview about GTEx. These are again, programs that have large epigenomic data sets or things that are relevant to that. And then I'm going to kind of switch and give you an overview on NIEHS, and what that stands for is the National Institute of Environmental Health Sciences, and our approach to supporting environmental exposure, studying environmental exposures and their influences on the epigenome. And I'll try to give you also some vignettes about some of the research that we actually support. Okay, I think I'm good. Okay. I work at the NIEHS in North Carolina, and it's our mission to reduce the burden of the environment on human health by discovering how exposures from the environment affect human health. We support research, training, outreach activities that encompass a broad and diverse set of approaches to address the the relationship of exposure to disease, development, and progression. The study of how environmental exposures impact epigenomic processes and marks in relationship to disease is certainly one of our research priorities at the NIEHS. So why do we want to study the epigenome? Epigenomics has certainly captured the headlines in both popular and scientific press in recent years. The plasticity of the epigenome makes it an attractable target for intervention and prevention strategies, and we think that the epigenome really serves as kind of an interface between the environment and the genome and the development of complex human diseases. The epigenome allows external signals or stimuli, or they can come from the internal as well. Chemicals are chemicals regardless of where they come from, whether they're endogenous or whether they come from the external environment. And it takes those stimuli and tells the genome what to do. Of course, you probably already know all this. Epigenomic changes have been implicated in a wide variety of human diseases. They're involved in normal processes of growth and development, and environmental exposures, which can include nutrition, chemical toxins, metals, psychosocial stressors, infections, drugs of abuse, as well as pharmaceuticals. These toxic exposures may result in a number of adverse health outcomes, including cardiovascular disease, autoimmune disorders, neurodevelopmental, as well as neurodegenerative disorders, and certainly, okay. And some of the environmental toxicants that we're so interested in that are associated with epigenetic changes in diseases are things that have been supported by the NIH for a number of years. We're interested in endocrine disrupting chemicals. That would include things like BPA, genistein, phthalates. We're interested in pesticides and herbicides, persistent organic pollutants. We're interested in metals such as lead, arsenic, chromium-6, nickel, and air pollutants that can come from diesel exhaust or particles. Now, the explosive growth in technology and development of next generation sequencing have really moved the field forward quickly, enabling high throughput interrogation of the epigenome in ways that just weren't possible a few years ago. Now, investigators are able to study various epigenomic marks, including DNA methylation, using a variety of different platforms, chromatin accessibility, and use ChIP-seq for protocols for histone modifications, and using RNA-seq to do functional readouts for expression, or look at non-coding RNAs such as link RNAs, microRNAs, bioRNAs. These are powerful methods which allow the investigation of the genome-wide distribution of methylation marks, chromatin binding proteins, histone modifications, and chromatin accessibility. And the data that's been produced by these next-generation sequencing technologies has also required the development of new and sophisticated software analytic packages and data analysis pipelines that help with the analysis of this huge volume of data. Okay, so this explosion of technology has allowed for the generation of unique data sets, and I want to talk to you a little bit about some of them, which would include, I will give you an overview of Roadmap, the International Human Epigenome Consortium, a little bit about ENCODE, something called GTEx, which is a gene tissue expression portal, and something about the 4D nucleome. I imagine you probably heard some of these things again over the last 10 days, and I'll just give you some overview before I go into stuff that we do at NIH. So what is the NIH Roadmap Epigenomics Program? The NIH Common Fund, formerly known as the Roadmap Program, supports big research opportunities that can create resources that change how we do science, and are supported by the Common Fund because there is no one single NIH institute that would be able to support the scope and breadth of the research that's being supported. There are 27 NIH institutes. Since 2008, the Roadmap Epigenomics Program has supported research in epigenomics with a primary goal of transforming our understanding of how epigenetics contributes to disease. The program supported several components, and the major piece was the Reference Epigenome Mapping Consortium, which again I think you've heard lots about. The goal of the Reference Epigenomic Mapping Consortium was to generate a community resource of epigenomic data in primary human cells and tissues. This is a publicly accessible resource for the community, and the data can be viewed and uploaded from a number of different sources. Other components within the roadmap program included the technology development, which really focused on imaging and single-cell epigenomic analyses, novel epigenomic marks discovery, projects on epigenomics of human health and disease, functional epigenomics, which basically support manipulation or editing of the epigenome primarily by CRISPR-Cas9, and computational analysis of the epigenome data sets that have been generated by the Reference Epigenome Mapping Centers. Before I go a little further, I just want to say one of the things that's really kind of some late breaking news with regards to technology development and imaging was just yesterday. Jacob Hooker at the Mass Gen and his colleagues published a paper called Insights into Neuroepigenetics through Human Histone Deacetylase, PET Imaging, in Science Translational Medicine. And what they've done is really fascinating. If you think about histone deacetylases, they're part of the epigenetic machinery that regulates gene transcription. In neurological disorders, HDACs change expression in regions throughout the brain, but their dynamic contribution to human disease development over time is unknown. Hooker and his colleagues therefore developed and applied an HDAC imaging probe called martenosat to visualize HDAC expression in the living brain. This is something that actually is able to cross the blood-brain barrier and then binds to HDACs. It was previously tested in rodents and non-human primates, and in this paper they report for the very first time they can look in living human brains. This is really exciting because it's going to really help look at neurodegenerative diseases. They engaged the HDACs that martenostat engaged with regulate the downstream genes that are important for neuroplasticity, memory, and neurodegeneration. So this is going to be really valuable tool, and that's kind of just an aside from our point, because someone just sent me this paper this morning. It's really exciting. So let's talk a little bit just about the Reference Epigenome Mapping Consortium, which again, you know something about, we'll probably talk about this a bit. The mapping centers were located at the Broad Institute in Boston. It was headed by Brad Bernstein and Alex Meissner. Joe Costello headed one at the University of California, San Francisco. Bing Ren headed the center at the University of California, San Diego. And John Stanatoyanopoulos headed one at the University of Washington. And we had also an epigenomics data analysis and coordination center that Alex Milosevic at Baylor College of Medicine headed. Okay, the Reference Epigenome Mapping Consortium produced reference epigenome maps from over 100 primary human tissues and cells, human embryonic stem cells, as well as induced pluripotent stem cells, and differentiated derivatives of human embryonic stem cells. This includes tissues and cells from pathologically normal adult donors, as well as fetal tissues. The fetal tissues came from elective pregnancy terminations. Samples were mapped both broadly and deeply. All of the maps have methylation data. We originally started out using different platforms for methylation, but as cost came down and technologies evolved, we switched to doing the maps with whole genome bisulfate sequencing. All of the maps have at least 5 to 6 core histone modifications mapped, and there's a smaller subset that has as many as 20 different histone modifications mapped. Many of them have chromatin accessibility data available, as well as gene expression data as functional readout, either by microarray tiling or RNA-seq. So again, we said this is a publicly available data set. Where are the data? The funding for the mapping consortium is basically ended. There are actually a few more data sets that are going to be generated from pathologically normal human brains. They've been slow coming in, but we are still doing that, and those data sets will be submitted directly to ENCODE. We can find these data sets available from You can get it from gene browsers, genome browsers at the WashU that have been maintained and developed by Ting Wang. And I would say Ting Wang and Professor Yu have been going around various different meetings around the country and internationally as well to provide workshops so people can use this data. Everybody that wants to use the data is really not what you call a hardcore epigenomicist or epigenetics person. And so we want the data, again, to be widely available and broadly used. So they've been conducting workshops all around and attracted, I guess, we had a big meeting called SOT, Society of Toxicology in New Orleans this past spring, and I guess you had probably 150 people there. And, I know you all sent me pictures from what you did in Whistler, British Columbia, and another meeting. So we're getting a lot of say dissemination of ways to use the data. So we have workshops. They've also created tutorials on how to do this. Another place that you can get this data is from the I don't know how that happened. Okay. It should. Okay. Okay, so anyway, I was saying where you can get these data. I said you can get them from the all right. The roadmap experiments have actually been migrated over to the ENCODE data portal, and you can access them from there. That's where they will be maintained, again, because the roadmap mapping consortium funding is pretty much run out. You can also get these also get these from the International Human Epigenome Consortium data portal, which basically has, this is more than the roadmap, but the roadmap data set, I can't really see that. That's, we have a number of different data, well, almost 3,000 data sets at the IHEC data portal. Okay, so I'll stop talking about roadmap now, and I'll talk a little about ENCODE. Another program that's developing large epigenomic data sets is ENCODE. That's funded by the National Human Genome Research Institute. It's an international collaboration of research groups with the goal of building a comprehensive list of functional elements in the human genome, including elements that act at the protein and RNA levels, regulatory elements that control cells and circumstances in which a gene is active. ENCODE investigators employ a variety of assays and methods to identify functional elements. Discovery and annotation of gene elements is accomplished primarily by sequencing a diverse range of RNA sequence sources, comparative genomics, integrated bioinformatic methods, and human curation. Regulatory elements are typically investigated through DNA hypersensitive assays, assays of DNA methylation and immunoprecipitation of proteins that interact with DNA and RNA, such as modified histones, transcription factors, chromatin regulators, and RNA binding proteins, followed by sequencing. All the ENCODE data is freely available for download and analysis, and there are again multiple tutorials available on their website for how you might go about it. And again, Fung has been responsible for, again, providing workshops around the country to actually do this, or teach people how to use this data. Push this. Okay, one of the things that an example of what we've learned from ENCODE is that the epigenomic modifications can mark functional genomic elements like enhancers and promoters, as well as transcriptionally active and silent regions of the genome. Okay, I'll talk a little bit about IHEC. As I warned you, I will give you an overview of the International Human Epigenome Consortium, or IHEC, which aims to coordinate epigenome mapping for a broad spectrum of human cell types at a wide range of developmental stages. IHEC is setting quality standards and providing recommendations for epigenomic analysis that the individual different national research enterprises that feed IHEC are creating. And the goal of the IHEC is really to produce 1,000 reference epigenomes. And with the data portal that we have, you can, again, access this, and we intend for rapid data sharing. There's a large multinational effort that's coordinated beyond what the United States has provided that totals $125 million US dollars. That's in excess of the $90 million that comes from the roadmap program, as well as the support that's coming from ENCODE as well. Data can be obtained through the IHEC data portal, and tracks can be viewed by Consortium. You can get the tracks either by looking at the tissues, or you can do it by assay, whether you're looking for transcription factor binding sites, methalomes, or regulomes, or histone modifications. Okay, I want to talk a little bit about what the member programs or the contributors to IHEC are. One of them is CREST, which is a Japanese project. It generates reference epigenomes from gastrointestinal epithelial cells, vascular endothelial cells, and cells of reproductive organs. The Blueprint program is supported by the European Union. It's generated at least 100 reference epigenomes of distinct types of hematopoietic cells from healthy individuals, as well as their malignant leukemic counterparts. The DEEP Consortium is supported by the German government and is developing maps from tissues and cells that are associated with metabolic and inflammatory diseases, such as adipocytes, liver cells, T cells, monocytes, macrophages, fibroblasts. It aims to generate at least 80 reference epigenomes. There's a Canadian epigenome project that's working on contributing 200 reference epigenomes that come from pathologically normal individuals, as well as cancer patients, patients with cardiovascular disease, and neurodegenerative disorders. There are also components and contributions to the IHEC from groups in Hong Kong, South Korea, and Singapore. Okay, I'll tell you a little bit about the 4D nucleome. The NIH, if you think about this, we spent about 15 years generating, developing the human DNA or genome sequence the human genome. The next 10 years really devoted to the roadmap epigenome program, and we developed a number of reference epigenome maps and technologies and this type of thing we talked about a little bit earlier. The next stage really to follow up on is the 4D nucleome. Okay, we know now that the DNA is not randomly arranged within the nucleus, and the organization of the nucleus is tightly controlled. However, the functional consequences of this organization are not well understood. What are the principles that govern the three-dimensional architecture of the nucleus? And how does this architecture contribute to gene expression regulation? How does the nuclear architecture change over time? That would be the fourth dimension when we talk about the 4D nucleome is time. Do dysfunctional alterations in nuclear organization lead to disease? Or could they be used to diagnose disease? The Common Fund's 4D nucleome program aims to understand the principles underlying nuclear organization in time and space, and the role that nuclear organization plays in gene expression and cellular function, and how these changes can affect normal development as well as various diseases. The program is developing technologies, resources, and data to enable the study of 4D nucleome, including novel tools to explore the dynamic nuclear architecture and its role in gene expression programs. Developing models to examine the relationship between nuclear organization and function in both normal development and disease, and developing reference maps of nuclear architecture in a variety of cells and tissues. For scientists to fully appreciate the dynamics of nuclear organization, it would be important to combine population-based models with data on individual cells at high resolution. This is going to require a multidisciplinary approach, bringing together genomics, biophysics, and imaging. The 4D nucleome program, recently funded by the NIH, is one initiative to seek to combine such expertise and includes consortia to map genomic structure, apply high-resolution imaging, and the properties of subnuclear compartments. The goals for tool development and improvement include improved experimental and computational methods for single-cell nucleome resolution and for the visualization of genome folding. Once the mapping of nuclear architecture becomes a more routine quantitative procedure, we'll be better able to determine and predict the effects of mutations, whether linked to disease or genome editing, on gene regulation and the mechanisms by which they act. We may even be able to specifically alter genome architecture to bring about desired changes within the cell. And just briefly talk about, in this section, just an overview of the genotype tissue expression, or GTEx program. GTEx projects are studying human gene expression and regulation in multiple tissues from healthy individuals. We've got over 960 post-mortem donors. They're healthy except for today. Expression data can be obtained from the GTEx portal, and the latest release in October 2015 had data from 450 donors with over 9,600 RNA-seq data sets across 51 different tissues. And this data can be viewed in multiple ways. You can look at the browse ETL tissues. You can look at the ETL visualizer. And you can also do the expression by tissues. It's an online data resource, and it's developing novel statistical methods for gene expression analysis, and it has a data coordinating center, and there are enhanced GTEx products that include additional dimensions beyond gene expression to the GTEx data. Okay, so all of these programs really are looking at normal tissues. And that's important, but I work for an agency that is really concerned with environmental exposures. So what does all of that mean to a toxicologist? I'm going to spend this part of my presentation discussing some of the programs that the NIEHS supports that are relevant to this, and provide some examples from projects that we support. Again, back in the beginning, I said that we look at the epigenome as an interface between the environment and the genome. And recently, the field has also begun to look at this. Exposures can alter gene expression by interaction with coding regions of the genome, but another important level of regulating or dysregulating gene expression is through exposure-induced modification of epigenomic marks and chromatin signatures. We need to know more about how the epigenome responds to environmental challenges by changing patterns of gene expression and how dependent those changes are on the underlying DNA sequence, and also what changes are robust enough to occur on diverse genetic backgrounds. And I just wanted to give you a little bit of, again, background for what NIEHS contributions to epigenetics and epigenomics have been. We established a program on epigenomic transgenerational inheritance that we call TIME, and that again is looking at how the epigenome, or how exposures, or how you can see traits that are induced by exposures seen in subsequent generations that have not seen that exposure. So we basically look at the F3 offspring because they have not been exposed, and we're looking at models where you can actually see the phenotypic traits that are attributed or ascribed to those exposures, but these animals were never exposed. We also do something called Target 1 and 2, which really is looking at chromatin structure, genomics, and transcriptional responses to the environment. And we supported a number of investigators that have really contributed to a really dramatic progression of papers or manuscripts published per year that look at how exposures actually impact the epigenome. Okay, so I want to tell you a little bit about the program we call Target, and it's called Toxin Exposures and Responsive by Genomic and Epigenomic Regulators of Transcription. It's a multi-phased program with 4 components. Phase 1 asks how do exposures actually lead to epigenetic and expression changes that we see. We have Phase 2, which is similar to the roadmap program, except from a toxicologist perspective, it's going to be better. Because we're actually using exposures. We use exposed rodent models and a variety of exposures, looking at a variety of different tissues. We engage a data coordinating center. And the questions that we're really trying to ask are, what changes are influenced by exposures? How do they change? And are the changes the same in all of the tissues if you expose them to whole animals? Okay, so as I said, this is a multiphasic program with Target 1, 2, 3, 4. Target 1 grants consider the impact of exposure on the following processes: nucleosome positioning, chromatin accessibility, chromatin modeling, the role of non-coding RNAs, and readers, writers, and erasers of epigenetic code. And we also are asking with these grants, how does underlying DNA sequence influence the impact of the exposures on epigenetic marks? We're currently supporting over 20 research grants in this area that have been awarded since 2013. And then Target 2 is focusing, and just recently been established, we just made awards in July, to develop a consortium that's really addressing a specific research gap and opportunity. There are currently a large number of human cohorts that have collected bloods, exfoliated cells from multiple sources, and other tissue cells, specimens that are available that have exposure data. And these are kind of what you would call surrogate tissues, but the investigators really don't know if what they're looking for in surrogate tissues like blood reflects what you're seeing in a tissue of interest where disease is happening, like a lung or brain tissue or something like that. And Target 2 is really being established to give us a better sense of what we can use, or what types of cells we can use or tissues that can be obtained using minimally invasive procedures to understand what is happening to the epigenomic landscape of disease-involved tissues. Target 2 focuses on identifying epigenomic signatures in mouse tissues exposed to environmental challenges, and also to ascertain if the changes observed in target tissues are conserved or correlative in surrogates. This is being done strictly in mouse models and is meant to inform something about how we might conduct similar studies with humans down the road. Okay, we have target 3 and 4 listed on this timeline, and they're going to be devoted to really looking at how we do population-based studies. Their projected time is on this timeline for implementation, but it's not really etched in stone because, again, we've just awarded the target 2. We got that going. So this is something that we're planning on down the road. Okay, so for this target 2, and I know that 2 comes after 1, but I'm going to talk about target 2 before I talk about target 1. The literature reports investigators using surrogate tissues, blood cells, and analyses of methylation signatures to get an insight on methylation signatures of breast cancer cells, adipose tissue, brain. But of course, none of these studies that appear were done in an exposure context. This consortium's work, Target 2, will give us a much better perspective on which types of exposures and surrogate target tissue pairs and under what conditions we'll be able to utilize surrogate tissue analysis of epigenomic signatures that are induced by environmental exposures. Okay, so to give you an overview of the Target 2 Consortium, we have selected and awarded investigators who are going to prioritize exposures, expose mice at different stages of life cycle. They're going to identify a number of what we call target tissues, as well as these surrogate pairs. They will apply a number of epigenomic and transcriptomic analyses that will then feed into a data coordinating center. And the goal of that data coordinating center will be to develop a publicly accessible database and resource for the community. The Target 2 Consortium is meant to generate a community resource, much like the Roadmap Epigenome Mapping Consortium or the ENCODE, but importantly, with an exposure context. There's no other large epigenomic consortium that is looking at anything with environmental exposure focus. Another key objective of this program is to really inform future population-based studies. The consortium consists of PIs from production grants, the PI of the Data Coordinating Center, and NIH program staff. Okay, and there's a charge to the Target 2. As this consortium is really in its nascent stage and just developing, there are a few things we need to keep in mind, and that would be what are the most important chemical exposures to prioritize, and how can the consortium inform future population-based research. The goal of this program is to develop a community resource that can provide some insight and perspective on how epigenomic signatures are changed after toxin challenge to whole animals. Okay, so there were a number of exposures that were originally proposed by the consortium membership, and they included endocrine disrupting chemicals such as BPA, genistein, and tributyl-10. They include phthalates, air pollution in the form of PM2.5, as well as dioxin. These are some great choices and represent some of the most studied chemical exposures by NIEHS-supported investigators, but it doesn't cover another priority class of chemicals or toxins, which are metals. So is there something we're missing? I just said yes, we're missing metals. And which metals are we missing? And things that are very high priority to us are arsenic and lead. So we have now added those chemicals after we made these awards in July, but prior to July we actually had a meeting and we've added these chemicals because they're again very high priority, or these metals because they're very high priority to us. So I want to give you again, this is a table that shows you the target and surrogate pairs that have been proposed by the different consortium memberships. This is Gokhan Mutlu at the University of Chicago. This is Shayan Biswal at the University of, Johns Hopkins University, and Sanjay Rajagopal at the University of Maryland. Cheryl Walker at the Texas A&M Health Sciences Center, has partnered with Marisa Bartolome at the University of Pennsylvania, and David Ayler at North Carolina State University, and David Dalinoy at the University of Michigan. As you can see, we've got a number of different tissue types that are targets, a number of surrogates, and these are exposures that we are going to be doing with this consortium. Okay, we have a data coordinating center that is led by Ting Wang at WashU. This is really important because one of the key things that we want to do is make this data publicly accessible. That's a primary reason for establishing the consortium. And I'd like to spend a minute or so just highlighting what the data coordinating center functions are. The role of the target data coordinating center will be to collect, store, curate, and display all data, metadata, and analysis tools that are generated by the consortium, and to make these resources available to the broader scientific community. The DCC is really the linchpin of this consortium and will provide several vital functions for the consortium, including generating a data submission pipeline, establishing a sustainable database, developing a public data portal, facilitating consortium communications through a variety of electronic media, conducting integrated analyses, and they'll be responsible for developing and performing outreach activities. And this outreach component is really a vital part as we will want to foster and encourage broader utilization of the data sets as they're being generated. Okay, so like I said, I know that 2 comes after 1, so I'm going to go back now and get the target 1. And this program was developed, we initially funded grants in 2012 to consider the impact of the environment on epigenomic and epigenetic processes in addition to methylation. At the time we started this, about 70% of what we funded with regards to epigenetics at NIH looked at methylation. And we know that methylation is very important, but there are certainly other epigenetic processes that we wanted to look at. And so target 1 is really meant to elucidate what happens to these other epigenomic marks in response to environmental stressors. As I said, we've got over 20 that are funded now, and we're looking at what's happening, what we consider to be upstream of DNA methylation. If you think about DNA methylation kind of as an endpoint to some things that happen. We're looking at a number of different exposures, including arsenic, nickel, chromium-6, endocrine disrupting chemicals, organic pollutants like 1,3-butadiene, and allergens like house dust mites, and also occupational exposures like hydrogen sulfide. We are also looking at readers, writers, and erasures of epigenetic code, and again, how chemical exposures can affect those and their function. Okay, so I'll spend the next few minutes talking about a few of these projects that we're funding. First, I'll talk about a project that uses house dust mite allergens and how this exposure interacts with the epigenome and are associated with asthma in both rodent models and lung cells taken from human asthmatic patients. Then I will go on to give you a little snapshot of something with nickel exposure and how that impacts the epigenome, and if time permits, then I'll talk a little bit about some arsenic exposures. Okay, Wanyi Tang at Johns Hopkins School of Medicine has a project looking at TET1 mediated DNA hydroxymethylation and airway hyperresponsiveness. And asthma is a complex disease that's characterized by airway hyperresponsiveness, airway inflammation, and it currently afflicts over 300 million people worldwide. Although genetic factors are unquestionably playing a role in asthma, the rapid rise in asthma prevalence around the world suggests that an environmental component or factor is equally important. There are a number of exposures that are associated with asthma and asthma attacks that include air pollution, high-fat diets, house dust mite, animal dander, and cigarette smoke. Normal airways tend to look like this. An asthmatic airway looks like this, where you see the walls inflamed and thickened, and then during an asthmatic attack, you have an increased response by
response to various different triggers and cause difficulties in breathing. Epigenetics may mediate the complex gene-environment interactions that influence asthma risk. And a person's epigenetics, which includes their histones, modifications, non-coding RNAs, and DNA methylations, can be modified after conception. There are people that may be born with genetic polymorphisms that predispose to asthma susceptibility, and there are clearly environmental factors that are associated. Epigenetic regulation is suggested to mediate at least partly the complex gene by environment interactions that can lead to asthma. And most studies to date have really focused on DNA methylation patterns in easily accessible cells like T cells and B cells to explain the immunological mechanisms that are driving allergen sensitization asthmatics. Few studies have actually examined the epigenetic consequences of environmental triggers on lung tissues linked to chronic alterations in lung function. Just, again, I'm not going to spend any time on this slide, but as you know, TET protein is involved in a passive demethylation by converting 5-methylcytosine to hydroxymethylcytosine into a passive demethylation and gene activation as a result of TET activation. TET requires for activity alpha-ketoglutarate, which is TCA intermediate, and Dr. Tang's hypothesis that she's testing is that house dust mite allergens activate TET1-mediated hydroxymethylation in airway smooth muscle cells through a regulation of redox recycling. This epigenetic reprogramming persists in airway smooth muscle cells over long time spans, likely contributing to increased airway smooth muscle cell proliferation, stiffness, and contractility that is seen in asthma patients. So she's got 3 aims that she's looking at, and the first aim of her project is to determine the role of TET1-mediated hydroxymethylation in the development of the airway hyperresponsive phenotype, which is increased airway reactivity, airway smooth muscle cell proliferation, contractility, and collagen deposition in the allergen-induced experimental model that she used with her mice. And when she's used TET1 knockout mice, her results clearly demonstrate that TET1 deficiency could protect the mice from increased airway hyperresponsiveness. You can see that when they have the she was able to protect these with the TET1 knockout. You can see that this response goes down. It goes down both in the lung tissues as well as the smooth airway smooth muscle cells. It's much more pronounced in these smooth muscle airway cells than it is in lung tissue, but it does offer some protection. Her second and third aims really look to examine if the regulation of the redox cycling and TCA cycle can modulate the TET1-mediated hydroxymethylation and ultimately reverse the lung cell functions of both the mouse and human asthmatics. So the second aim really looks at sulforaphane, which can reverse redox or remove ROS in the alpha-ketoglutarate, which can inhibit the activation of TET and effectively keep the 5-methylcytosine present and doesn't activate TGF-beta signaling genes that are associated with smooth airway muscle hyperresponsiveness. And her results showed that the sulforaphane actually did reverse the hypersensitive, hyperresponsive from the house dust mites. It reduces the 5-hydroxymethylcytosine level, it reduces the TET activity, as well as reducing the alpha-ketoglutarate levels. And the aim 3 was really to investigate the role of TET1-mediated DNA hydroxymethylation in regulating asthma phenotypes in human cells. And and they got cells from asthmatic patients and non-asthmatic patients from commercially available sources, and they knocked down TET1 with SI-TET1, and again were able to effectively reduce or revert in the asthmatic patients that phenotype. Okay, I can talk a little about nickel, but I think at this point I just want to show you without going through data, show you kind of using lung cells that were exposed to nickel. They examined. They examined the effect on several histone modifications using ChIP-seq and showed that H3K9 dimethylation is significantly altered by nickel exposure, resulting in gene silencing. I won't bother to go through this data at this point. And I would also show you that basically the key findings from this study are the H3K9 dimethylation forms domain structures are depleted at active gene promoters. They find that CTCF delineates H3K9 dimethylation domains. There's a spreading of K9 dimethylation domains that are displaced from CTCF on nickel exposure, and that the H3K9 dimethylation spreading is associated with an aberrant gene size. And there are tumor suppressor genes that are among those aberrantly silenced by the spreading that are involved in nickel-associated carcinogenesis. I think, I'm not sure what my time is, but I think that I will probably stop at this point. If there are questions that anybody has, I'd be happy to try to answer those.
That's fine. I think thank you, Mike. So Fred, thanks for giving us such a comprehensive review of the ongoing efforts of the NIH, Fred NIH, EHS. You notice now the hottest word is precision medicine.
What's that?
The hottest word now is precision medicine.
Yes.
Right. So, these young people, they are interested in academics. They also want to work on precision medicine. So, any comments on that field? The academics now precision medicine, how can it affect the community, diagnosis, so on and so forth?
My comments would be on that. Obviously, NIH is developing a precision medicine initiative, and I think one of the real drivers in that is going to be how we can better develop single-cell epigenomic analyses, and how we can also look at individuals' genotypes, because certainly what we're seeing in some mouse studies, when we look at environmental exposures, like one example I could give you would be with 1,3-butadiene, depending on the genetic background, if you use a black 6 mouse versus a different strain that's being used, you can see where you can get some methylation changes, or you can get some changes in link RNA functions that really are dependent on genotypes. So I think being able to look at people's genotypes, as well as doing some single-cell analysis or improving that, is going to really help us. Also, the thing that's going to help with that is imaging. And we talked about, when I mentioned yesterday's report from Jacob Hooker, in terms of being able to look in living tissues or living people at tissues, I think that's also going to be a big contributor. So being able to do the single-cell resolution, being able to do imaging in live patients, as well as taking into account individual genotypes, is going to be important in developing precision medicine initiatives.
Great. So another perspective, or the things I want to learn from you, is there are quite a few medical doctors in this room, and then there's a basic scientist who want to get into the medical side as well? Like from your personal experience or your administrative experience, what are the potential obstacles for the basic scientists working with medical doctors to push a collaboration or push a project forward more effectively?
That's an interesting question in terms of what are impediments. I don't know what impediments are, other than availability of funding. Within our program, within the roadmap, for instance, we had several physician scientists that collaborated with basic scientists that included certainly Brad Bernstein and John Stam. And I don't know what the impediments are short of funding.
Hi, Fred. I know you have a lot of expertise in this kind of project management, right? So my question is when you deal with some very big project, there's maybe some like INCO project or roadmap project. They must have some three statements, right? Initiation or like project design process or ongoing process. So my question is, when you design a project or when you start up a new project, because you are leading the cutting edge technology, because you are leading the direction of all science, right? So when you start up a project, will you use the top-down plan or bottom-up? Will you use a big vision to just ignore any just not care about details, or you use a very specific hypothesis, some specific signal will induce or kind of change, and this kind of hypothesis to do is kind of bottom-up. So what's your bigger vision and debating with small details?
Okay, I'm not quite sure I understand your question. If I understand it correctly, you're asking in terms of developing these larger types of programs, is it basically a top-down? I guess by a top-down you mean the funding agency in terms of giving direction for the program? Okay. The way let me explain actually, for instance, the roadmap program is a really good example of how we do this. To develop the roadmap program, first of all, at that time in 2000, actually we started that process in 2006 and actually made awards in 2008. What happened was we had 150 nominations for what should be a big ticket project that came into the NIH. They were submitted by scientists in the field as well as by NIH program staff like myself. That went to a selection of people much higher up than me that kind of boiled that down to 8. And what those 8 opportunities, what those 8 potential programs did was we had we're given some money, some seed money, to really bring in a group of experts. And so for us to develop the roadmap program, we brought in 30 different people from around the world, not just within the U.S., but we brought in investigators from around the world to give us some perspective in terms of telling us what was needed to move the field forward. And we met for 3 days, and we got a series of recommendations that came from scientists, not from program staff like myself, but from people in the field that gave us these different recommendations that once we had that, we developed a pilot program that we then competed again among those last 8 that were standing. And then 2 were selected. Roadmap, the epigenomics was one that was remaining. I don't remember the other one that came, or maybe it was the microbiome. But we actually get our information in terms of what gaps we want to address from the field. Now, and how we actually manage that is different. But we actually really solicit the field. It's not, we have an idea, but we don't just run with that. We get guidance from the people who will actually do the work.
So there are a pilot study or validation, and those 2 bigger and bigger, right?
Yeah, well, the piloting is actually, we develop what we want to do and we present that, and then we either get a green light to do it or not. So we're not actually doing any studies other than we're crafting the program. We get a lot of input from a lot of different places
before we actually put a solicitation on the street. And then when actually applications get scored, then we kind of pick and see what is best fitting the niches that we've identified through consult with people who are in the field.
Thanks, Fred, again. So the next few minutes, we are going to do the award ceremony. So as you know, I didn't know this, but apparently many people voted on the WeChat, and the judges sitting in the front row, we also did our voting. Now the results come in, and let's see who win the big award. What's the award anyway? Is it cash? Yeah, we'll see. The award is taking a picture with the professor. How do we do this? Do we need to? Okay, so I think we want to make the process transparent. So there are two categories. One is students' vote. Each student get one vote, but it's not completely democratic because professors got the vote is much heavier, right? I think one person's vote counts for 10, 20, something like that, right?
And the student vote before 1:30. After 1:30.
Okay. Okay, yeah. So it's apparent. We will have 3 classes of award. We have 3 third place award, right? And 2 second place award and 1 first place award. And now let's announce the, and then, let's go with the number 3. Oh, next slide, okay. Final ranking. First place, no. Why do we shorten everything? There's no drama. Okay, the first place is Jing Li, which is not surprising, from RG Daxue. Congratulations, Jing. Second prize, Yan Xia from Zhongnan Daxue, and Liu Fa Yang from Xibei Nongli. Congratulations. Third place is that guy who studied the relationship between academic pressure and hair loss, Xu Peng from Fudan University. And Hao Ya Juan from academic science. Last person is Wei Jin from University of Hong Kong. Congratulations to everyone. You are young and you're creative, and you'll achieve great success in the very near future. Keep it up. And we will do the award ceremony now. Yeah. So for the judges, I think each of us will give the prize to the prize winner and take a picture. Give us your biggest smile, and I'm sure that picture will be somewhere on the website. All right, we'll start from the third class prize.